Okay, so we've taken a look at how we discovered that DNA was the genetic material, how we cracked the codon table, and were able to find out which codons were responsible for coding for which amino acids, and we talked about how we knew that prokaryotes used plasmids to transfer DNA or traits. Now we're going to go a bit more into how alterations to the DNA and to the genome are able to influence phenotypes. So it's going a bit more into depth on the genetic side of things. So the genome is obviously disposed to mutations which alter the genome, alter the nucleotides, and there are many different kinds of mutations that can occur, the first of which are deletions, which are caused by a loss of DNA in the genome that can be either small or large and can result in the loss of entire genes caused by a process called replication slippage. So this would be this example at the bottom here, where this thymine, shown in blue, has been deleted, possibly due to a replication slippage when the DNA has replicated for cell division. So this cell, which would be a daughter cell of the top cell, doesn't have this thymine, so it's a shorter fragment of DNA. It's lost this single nucleotide. And this principle can apply to larger stretches of DNA and even to entire genes. An insertion is the opposite, where gain of a DNA sequence, so DNA can gain nucleotides. This can either be small or large and can result in whole genome duplications as well as whole gene duplications. These tend to be interspersed genomic repeats, and they're also usually caused by replication slippage. Substitutions are a bit different. This is where a base is replaced with another base at a specific locus. These substitutions can be synonymous if the change in base results in a codon that codes for the same amino acid, so it's not going to have any bearing, it's not going to change the polypeptide produced by that gene whatsoever. But then it could also be non-synonymous if the base pair change results in a different codon, which codes for a different amino acid, and that's subsequently going to alter the structure of the polypeptide. Usually this is only a minor change in structure that doesn't really affect much, but it should be noted that sometimes a single base pair substitution or insertion or deletion, sometimes that causing a single change to one amino acid can have pretty catastrophic effects, like we're going to talk to in a minute, on sickle cell anemia, which is caused by a single nucleotide polymorphism, which alters the hemoglobin protein because a single different amino acid is added, which changes the structure of the hemoglobin and results in red blood cells not being able to do what they're supposed to do, and gives them a kind of lower affinity for oxygen and a sickle shape. You also have rearrangements where DNA can be excised and reinserted into the genome, so taken out and put back in, but in the opposite orientation. This was famously what caused the white eyes in Morgan's white-eyed Drosophila, where the W gene that codes for the red pigment in the eyes was rearranged, so it was excised and reinserted in the fly's genome in the opposite orientation, and when it was rearranged, the W gene coding for the red pigment was placed in a transcriptionally repressed region of the genome that was not unwound from a nucleosome. So the red eye pigment could not be transcribed or translated, so none of the red eye pigment was produced, which is why the white-eyed Drosophila have white eyes. So, point mutations refer to single base pair changes. Any of these can be point mutations, deletions, insertions, substitutions. I don't think rearrangements can be, but these three can all be point mutations, and you also have gross or large-scale mutations, which are these things but applied to larger structures of DNA. Neither have any effect if they occur in an intron, which is a non-coding region of the genome, or in a nucleosome-associated locus of the genome, because these regions are transcriptionally repressed. So, talking a bit more about the type of mutations that can occur, in missense mutations, a single base pair change can alter a single codon to produce a different amino acid. These are usually tolerated, but there are exceptions like we talked about a minute ago with sickle cell. A nonsense mutation, where a single base pair change can produce a premature stop codon in a gene, resulting in the gene product being truncated and typically dysfunctional, as is the case with Duchenne's muscular dystrophy shown on the bottom right. You can have a frame shift mutation, where an insertion or deletion can alter the triplet reading frame of the post-mRNA produced by a gene, resulting in a completely different protein being produced. So it doesn't really work for this example that I've put here, because they're all the exact same amino, all the exact same bases. But say we were to look at this one here, look at the very bottom strand of DNA, you can see CCGAA, CCGA. Let's say the first three, CCG code for one amino acid, AAC code for another amino acid, and then CGA code for another amino acid. Okay? If we were to have one of these kind of mutations and take one of these base pairs out, well, it wouldn't just affect that codon, because if you were to completely remove it, because at the ribosome bases are read in sequences of 3, being the codons, like we talked about here. If you were to, say, take out this first A here, so the first A to the left, if you were to do that, you'd have the exact same first amino acid being produced by the CCT codon, but then taking away that A, that would result in the second codon being ACC, because the reading frame, the amino acid, the ribosome is reading the codons and the bases in sequences of 3, and then the final piece of the mRNA would be read as GA, and then whatever is continuing to the right. So a frame shift mutation completely alters the reading frame, so it results in a completely different polypeptide being produced when the gene is translated at the ribosome. And then conversely, you have a silent mutation, which typically occurs at the third base pair in a codon, which results in the new codon basically coding for the exact same amino acid. So like we talked about when we talked about the codon chart, and how there are many different codons.